Today, I want to do three things with you. I want to just go back and review something that I put on the board last week. That guy's dead. And I also sent this to you via email yesterday evening, largely out of responses from questions I was getting last week from students. Then I want to provide more background on the Gecko data set so that you just kind of understand that data set a little more. And all of my black markers are dying. And then the next thing we're going to be doing in this class is two-way analysis of variance. And so I want to start going through that today. So for this first part, I sent out a Word document last night, I guess it was. And basically, this is just kind of how we want to go through and do an analysis. And if you graduate from William Jewell College and you become a data science type of person, and you're in an organization where they're asking you to do data analysis rather than just kind of data organization and data wrangling and database management and things like that, you have no idea what kind of project they're going to bring you to do an analysis of. And so you have to kind of have a system for going through and getting a handle on things. And so when somebody comes to me with an analysis that they need help on, the very first thing that I do is I sit down and I think I've told you guys about this before, I sit down and I schedule a meeting with them to basically get them to talk through what their data are about, what their project is about, what questions they're trying to answer, what their data are. And this works whether you're talking to a molecular biologist, whether you're talking to an ecologist, whether you're talking to a business person, whatever it happens to be. And you have to know enough about what they're doing that you can make wise statistical decisions. Hopefully, one of the things that you're learning, for example, by doing this project that you're working on for Wednesday, is that statistics isn't just a set of procedures that you step through like a mindless drone without any sort of critical thought being exercised towards the data. You actually do have to make decisions. Statistical analysis isn't kind of set in stone. You do X, then you do Y, then you do Z, and then you get the answer at the end. You have to make judgment calls periodically about the data that you're working with. And so in that way, developing some statistical judgment is actually important. So the easiest way to make good judgments is to understand as much about the person's data as you can from them. I mentioned that I was working with a group of students here in Dr. Bhupinder Vora's lab on a data analysis that they were working on, and it took us 3 one-hour meetings for them to get to the point where they were thinking about their data in terms of their questions in a way that was productive for me as a statistician to look at their data. And so it can be a time-consuming and rough process at the beginning. What I'm going to do with you after I'm done going through this thing is I'm going to show you kind of the broader picture of what this Gecko data came from, so you will have a more informed idea of what the researcher, i.e. me, is thinking about when I think about these data, because I think that will help inform you as you write your reports. And we didn't have that opportunity to sit down and have you pepper me with questions about the data set and things like this. So we'll do a little bit of that today. Once you're done with that, think about the analysis that you're going to do. As you think about the analysis that you're going to do, you can kind of foresee what packages you're going to need because you know what analysis it is that you're going to be using. And so install those packages up front, read in the data, check for errors. We've talked about this before. In this document, I've given you a variety of different commands that you can use for doing this checking. We've been through all of these in class, but here they are in one section where you can kind of find them all written down for you. Then checking the assumptions. And this is where I think some of you have gotten stuck. When you check assumptions, the assumptions that you're checking are, are the data normally distributed? And the other question that you were trying to ask is, are the variances among the treatments equal? If the data are normal, what's the p-value that you should expect to see out of a Shapiro-Wilk test? I find it shocking that some of you are still struggling with this, which is why I'm reviewing it. If the data are normally distributed, what p-value should you expect to see?
Greater than 0.05.
Greater than 0.05. So why did I put an equal sign there? If the variances are equal, what should you expect to see for a p-value? Greater than 0.05, yeah. If the variances are equal, it should be greater than 0.05. Because the test is...
What's the one where it needs to be, if it's less than significant?
None. We're talking p-values. So in the Shapiro-Wilk, the test statistic is flipped, but the p-value is always the same. What is the p-value again? Let's just go back and review. What is the p-value? It's the observed type 1 error rate for your particular data set. So the reason that we use p rather than using the Shapiro-Wilk test, or the Shapiro-Wilk statistic, or using the S statistic, or whatever, is p is universal. It's the thing that comes out of any kind of analysis, any test statistic that you might have. What is a type 1 error rate for comparing means to one another? Concluding means are what? Different when they are so we call it an error because it is an error. You conclude that means are different when they are not. For an Anova, it's concluding that at least two means are different when they are not.
And then for an Anova, if it's less than 0.05, then it's significant.
Yes, because you don't want to make this mistake very often. So this is a mistake. This is a mistake you want to avoid. So how often do you want to make this mistake? You don't want to make it any more than 5% of the time. So you're looking for a p-value of less than 0.05 before you say that things are different. What does this mean for a normal distribution? It's concluding non-normality when the distribution is normal. P-value. It's different for different tests because different tests are testing different things, but it's always concluding lack of equality when the truth is equality. Are these data distributed normally? No, they're not. Making that decision when that decision actually isn't true because they actually are normal. So in all these cases, you hope that your Shapiro-Wilk test has a p-value greater than 0.05 and your variances have a p-value greater than 0.05. If that's the case, you don't do anything. You just go on and go to the next step and do the analysis. On the other hand, if you get p-values that are less than 0.05, this means you have non-normal data, or you have heterogeneous variances rather than homogeneous variances. Then you have to start thinking. You have to start thinking critically about your data, because if you see that you have a significant deviation from normality, it might be that you just have a huge sample size, and so it's really easy to detect those differences from normality. If you don't see a significant difference, if you see this, it could just be that your sample sizes are so low that you can't adequately test that question. You end up with a p-value of 0.8, and you're like, woo, normal distributions. And then you look at the distribution, and you're like, that doesn't look normal. Why was my test not showing me that it's non-normal? Well, because you have really low sample sizes, and so you can't detect that non-normality very easily. So you have to think critically about the data. And the way to do this is to visualize the data. If you get a significant deviation from normality, make a histogram of the data. And if the histogram of the data looks pretty normal, then ask yourself, well, what's my sample size? For the Gecko dataset, what's the sample size for the normality test? (182). Is that a little bit of data or a lot of data? That's a lot of data. You can detect really tiny deviations from normality with 182 data points. It doesn't have to be very non-normal to pick that up with 182 data points. What was the p-value for that when you ran that? It's just right on the cusp of being significant. Like, it's just a teensy-weensy bit significant based on the p-value. But we have a sample size of 182. Our individual sample sizes for each treatment are n equals 30, 40, 52, and 60. Are those large sample sizes or small sample sizes? Those are relatively large, unless you're a genomic scientist, in which case they're tiny. But for most data sets, 30 is kind of the magical number, after which you can do what? Central limit theorem saves our butt because it says that the sample means, the distribution of sample means, is going to be normally distributed, so it doesn't really matter what the actual distribution of the population that the data came from looks like. The distribution of sample means you pull from that population will be normally distributed. Woo! Give it up for the central limit theorem. If you find that you have p is greater than 0.05 for homogeneity of variances, if you get a non-significant result, once again, it could be that your variances are different and you just don't have enough sample size to detect that they're different. That does not mean that you can just blithely go ahead and do your statistical test because you got a p-value of greater than 0.05. Because when you graph your data, make a histogram of your data, you might find that some of your data have distributions that are really broad, and some of your populations, some of your samples have distributions that are really narrow. They look like the Levine's test should be significant, but it's not, because you just don't have enough data to detect that difference. Now, if you do detect a difference and your distributions of your data look kind of like this, you will get a significant p-value that will show that you do have variances that are not homogeneous. Do you really need to worry about this in this case? Karis is shaking his head no. Somebody other than him, tell me why you don't really need to worry about this very much. What are those distributions not doing? They're not overlapping. There's no risk of you concluding that the mean that you generate from this population could have come from this population and vice versa. So even though the variances are different in this case, it doesn't pose as much of a problem as if they were overlapping a lot because of the reasons that we've now discussed what feels like 15 or 20 times now. So it could be that you find a non-significant Levine's test because you just have small sample sizes. Once again, you need to visualize the data and assess whether or not you should trust that result. In the same way, if you get a significant result, you should visualize those data and get a visual assessment of whether or not that's actually probably true and whether or not that's going to cause a problem for the Anova, because it could also be that you have a significant Levine's test simply because you have a really large sample size, and your textbook is really clear about this on page 193. Simply because you have a significant result of Levine's test telling you that your variances are not equal does not mean that that is going to destroy the results of your Anova. It depends on how big your sample size is, how different are those variances actually, and how do those variances line up with where the means are in terms of location. If they're not really overlapping, it's not as big of a problem. If they are overlapping, well, then you have some issues. And you need to then work through what the transformations would be for that. All of this requires you to be actively, thoughtfully engaged in the analysis. You can't just step through it like it's a series of steps, even though I'm realizing now I kind of give you a Word document that's just a series of steps. So you have to think about it. You have to think about it intelligently. And hopefully I've communicated to you the ways of thinking about this enough times that you can do this effectively. If you can't, then definitely come by and see me and I'll try and get it through again. If there is a problem, then you have to transform your data. Your textbook goes through all of the standard transformations. I gave you a very atypical transformation for the P's data set because that's the transformation that the textbook used in this textbook example, but that is not a typical transformation. Go to your textbook for those. Do you guys recall what the Levine's test p-value was? That is the p-value for Levine's test for the Gecko dataset. You have variances that are not equal. But once again, go and look at the histograms of those. Look at where the means lie within those histograms, et cetera, and ask yourself, is this simply a function because I have huge sample sizes because the variances are relatively small, and they're especially small relative to the differences among the means. Even the largest one is really small confidence interval relative to the distance between it and the means that it's different from. So what I did for this data set is I said, after I look at the data, that Levine's test result is probably a function of large sample size because the confidence intervals around the means don't have the problem of making ambiguous decisions between means. When the means are the same, when the means are determined to be equal in this data set, it's very clear that they are equal. And when the means are determined to be different in this data set, it's very clear that they are different. It's not ambiguous at all. And so I opted in my own analysis of these data to basically ignore this. But when you ignore it, you have to explain why. And it is a judgment call. And there are some people who will die on the hill of, if you get a significant Levine's test or you get a significant normality test, you have to do some sort of transformation on the data. I am not in that camp. The authors of your textbook are not in that camp. The authors of your textbook are about exercising good statistical judgment. Because here's the thing about real data, and these are real data. Me and Jacob Marley You guys don't know Jacob, do you know? He graduated before you guys got here, I think. Me and Jacob Marley gathered all of this data. Real world data, guess what? They are messy. They are not clean. They are not textbook data. If you guys are going out in the world of data science, just get ready for a lot of very messy, complicated, difficult to deal with data. It's just the way the world is. And so you have to get some statistical judgment in your head. All right. Once you've transformed the data, you have to go back and recheck all this stuff again. In all cases, you should inform your client of what you did and why you did it, or did nothing for that matter. Then you run the analysis, you find the result. If it looks like things are different, you either run planned, that should say planned comparisons, not planed comparisons. You run planned comparisons if the person had some a priori hypotheses about what might be different from what. And if they didn't, then you run post hoc comparisons, which are something like Tukey's test and an Anova, depending on what the overall Anova showed you at the beginning. You want to back transfer your data so that you can graph your data in meaningful terms if you had to transform your data. And then you could create meaningful tables and graphics. I'm a big fan of seeing the Anova table. If I'm reading a paper in the scientific literature and they give me an Anova table, I can look at that thing instantly and know whether or not they analyze their data correctly, because they've described what their experiment is like, they've described what their sample sizes were, and all I have to do is count up the degrees of freedom to know if they've messed up. And you will be surprised at how many people screw up their statistics, and it's detectable in an Anova table. Unfortunately, about 20 years ago, we stopped publishing Anova tables. I would like to see your Anova table because I can tell at a glance whether you did things correctly by looking at the numbers in your Anova table. Yes? Summary model. So if you set up your model, so if you made model, if you did this, all you have to do is type in summary model, and it'll give you the Anova result in an Anova table. It should be in the live coding scripts that we worked on. It's also in the P's analysis R markdown that I have posted on the Moodle site. So that's available to you.
Thank you.
Yeah, sure. Graphics should be inferential. What do I mean when I say graphics should be inferential? Some of you are looking like, how the heck should we know what you mean when you say that? What are we trying to do when we run an Anova? We're making an inference about what? We're making an inference about the means, right? We want to know if the means are different. So you should show your client graphs that show them whether or not means are different. How do we do that? Distributions are less helpful, actually. Because how do we determine if two means are different visually? No. Give it another shot. Yeah, you knew it. I thought you knew it. Good. You look at the confidence intervals and you see if one mean fits in the confidence interval of the other mean and vice versa. So you want to be graphing things in ways that you can just take a quick glance at the graph and tell whether or not means are actually different. You're going to run a Tukey's test or whatever on it, but you want to make sure that your graphic reinforces the results of the Tukey's test. If the Tukey's test says one thing and then you produce a graphic and it doesn't look like the results of the Tukey test, the client is going to be like, are you sure you did this right? And then you're going to be like, oh yeah, sorry about that. I graphed the standard deviation rather than the confidence interval. Trust me, it's actually different. But you don't have to have that conversation with your client if you graph the 95% confidence intervals from the very beginning. the graph ends up looking like what the Tukey's test says the graph should look like. A couple of notes about figure captions and table captions. They have a particular form. The form is here. They all have a number. They have a title, which is basically a description of what the graph is. And they all have then an explanation, which is an explanation of what the graph is saying and what that means. You will also usually see this in the body of your report, in the text of your report, because quite frankly, oftentimes clients aren't reading very carefully, and if you put it two places, they're more likely to get the message. And that's true for if you're doing this report for a client or you're writing something up for publication. It doesn't hurt to repeat yourself, because oftentimes that's the way things get across. There's a good idea in pedagogy that you have to tell students or expose students to something 5 times before they get it. Same thing might be applied to writing reports for clients that you might have. So then once you've done all that, all you have to do is sit down and write your report. As I've said before, the introduction should contain what you believe to be the task is at hand, which includes your understanding of what the data are and what the questions are that your client has about those data. A section on the statistical methods describing what you did and why you did it that way as clearly as you can, and then a results section that describes what you found as a result of those statistical machinations. And that's what you need to turn in. Rachel, you gave me a draft of yours. It ended up being what, about 2 pages with graphs and everything? 2 or 3 pages. These are not necessarily long reports, but they do need to have these components in them. The reports will get longer with each subsequent report because the analysis that we're going to be doing is more complicated and more involved. This first one is on the easy side. Once again, I provided you a template for how this report should look on Moodle in the P's analysis. So I've written up the P's analysis as if I was analyzing this for somebody and did all of these things in that analysis. So you can hopefully see what that is about. Questions about the report? I've hopefully now given you enough tools that you can do this. If you're having trouble with code running and things like that, then make an appointment, come by and see me, and we'll sort through that. Yeah.
Do it to start the class on Wednesday?
You want to start a class or end of the day? It doesn't matter to me because it.
End of the day.
Okay, end of the day, that's fine. It doesn't matter to me because what we do in class doesn't depend upon you having it done. Yeah, Rachel. I'm going to put links on the Moodle site. So save all of these things. You can save your Markdown file as an HTML file. So I will have a separate link for the our Markdown file, and I will. You're looking at me like you don't know what I mean when I say that. No, you're thinking of something else. So use the knitting option to knit it to either an HTML file, a PDF, or a Word file. Any of those work. Some of them you have to have extra software in order to do it. The easiest one to knit to is an HTML file. So I will have a link for submitting your R Markdown file, and I will have a link for you to submit your PDF of your report. And I would prefer to have it in PDF form because then I can just easily download it to my iPad and read it without printing out a paper copy of it. And I can mark on it that way also, yeah.
And just like one last time for the report, we're going to just use like a Word or
Yep. Just save it as a PDF. It just makes it easier on me. So there will be 2 links there, and then I'll actually grade them on my iPad, and then I'll send them back to you digitally with marks on it. Yeah.
You know, the visuals on the PDF and not the HTML?
It'll appear both places. So chances are you're going to generate a lot more graphics in your R Markdown file than you actually need to put into your report. So I'll see them in either place. The reason I want the R Markdown file is so that when I read your report and something looks wonky to me, I can go back to your R Markdown file and see what happened and why you generated the thing that you generated. So the nice thing about the R Markdown files is that they give me not only the code that you wrote, but it also gives me the output after you've written that code, which is why in your R Markdown file, it will make life easier on me that anytime you look at your entire data set by just typing its name, or anytime you head your data set or tail your data set. Well, I put the parentheses in the wrong place. But especially if you just look at the whole thing all at once, please comment those out in your R Markdown file. Otherwise, it's going to give me like 3 pages. It's just the data. And I have to scroll past that. And so clean up those things a little bit before you turn it in, just for my sanity. Yeah.
Is that the only thing you want us commenting out?
I think so. When you do structure and things like that, I'm assuming you're doing that because you just made a change and you're checking to see if R made the change that you think it made. And that's good statistical procedure, and I don't mind that. What I don't want is 182 lines taking up the space in your R Markdown file. You will notice in the P's analysis R Markdown file that is on Moodle, I did not do that. Scroll through that and see how irritating it is to be just subjected to big chunks of data when you're not expecting it. It's just an irritation, and I would rather not be irritable while I'm grading your paper. So that's that. Any other questions about the report? Hopefully you've all gotten started. It doesn't take a ton of time to do this if you're smooth and you're not encountering any problems. But if you start to encounter problems, it can take longer. Rachel's is the only one that I saw. How long did it take you to write the report actually? Well, yeah, but you have the framework is there. Yeah, it doesn't take long to write it up once you've done it all. All right. Other questions? I'm only asking you because you're the only one who has a draft that I've seen. All right. So let me give you a little more background about this whole gecko thing. So we're going to spend, every report that you do is going to be on the gecko analysis. And the reason that I do this is because there's a lot of different ways that you can analyze the gecko data set. And because it's all one kind of integrated project, you get to know it over the course of the semester. And so it kind of represents a large, complicated sort of data set, which is the kind of data set that you might be faced with out there in the real world. Seldom is somebody going to bring you a data set and it's all nice and tidy, tied up in a bow. It requires one particular statistical technique and then you're done. I've not had a data set like that since I was an undergraduate student. So data sets are complicated, and this is one of those things that is true for this particular data set. So I'm going to have the conversation that I would normally have with the researcher with you guys. And what I'm going to do is I'm going to present stuff to you. And as I'm presenting it, I want you to feel free to ask me questions as we go along so that you understand things. And this is what you should do anytime somebody brings a data set to you. So I'm interested in the behavioral ecology of amphibians and reptiles. I work with an introduced species here in the United States. And this introduced species was introduced into the United States back in the early part of the 20th century and has expanded throughout the United States. As it expands throughout the United States, it comes into contact with different thermal environments. So oftentimes when we think about temperature changing in the world, you hear a lot of talk about global warming, and that is a concern for people. But because we spend a lot of time talking about global warming, we don't talk about other types of temperature stress that organisms are faced with. And so in the last couple of years, this was something from 2018, here's a report from the New York Times talking about lizards falling out of trees in Florida. So here you have an iguana, which is a tropical species. Tropical species live in places where it's warm all the time. These things move into Florida, and Florida is pretty warm all the time, but occasionally you get these cold fronts that move all the way down into southern Florida, and at that point, these lizards are too cold, and they don't die from it, but they become immobilized by it. They get so cold that they can't move. And the reason that they get so cold that they can't move is because they don't generate their body heat through metabolism. You're sitting there, your brain is working, your body is using up calories, it's generating a ton of heat, and that's what keeps you warm. That's why you can go out in winter, do winter activities, go skiing and everything else, because you have a metabolism that keeps you warm. Lizards and snakes and turtles don't have a metabolism that keeps them warm, so their body temperature tends to fluctuate more with the climate that the animal finds itself in. And when you get cold enough, your physiology slows down enough that you lose the ability to actually walk around, or in this case, cling to a tree. So all these lizards were falling down into people's pools in southern Florida. They thought they were dead. They brought them in the house. They warmed up, and they came back to life. Well, they didn't come back to life. They were never dead to begin with. So when we think about introduced species, my particular introduced species is from southern Europe and northern Africa. It's called the Mediterranean gecko because it's from the Mediterranean region. This is the Mediterranean Sea here. And it initially got introduced into the southern United States, where it's warm, where temperatures are not that different from southern Europe and northern Africa. But then it started expanding throughout the United States, and some of this expansion took it northward. And so all of a sudden, this is what it looks like. All of a sudden, you have this warm species that has migrated north. There's now a population in Olathe, right in a suburb of Kansas City. There's also a population all the way up here in Pittsburgh. These are places that get cold. So you have this warm weather species, this warm climate species, that is now invading new environments that are much colder than the environment that it comes from. And I'm interested in how it's coping with that whole situation. So if you take a lizard and put it in a racetrack and measure its speed, if you take a cold lizard and put it in a racetrack, it doesn't run very fast at all. And if you warm that lizard up, it runs faster and faster and faster until it reaches some peak of performance. And then if you continue to warm up that lizard, it begins to slow down because you're basically killing it because it's getting too hot. And so it stops performing so well because it's beginning to die because its proteins are being denatured. Normally, lizards moderate their body temperature through behavior. So lizards will be out in the sun. They'll get nice and warm. 37 degrees is your body temperature, and it's common body temperature for lots of lizard species also in that 35 to 38 degree range. If they get too hot, they will run over and hang out in the shade and cool down. And when they get too cool, they'll run back in the sun. If they get really, really hot, they might go underneath a rock where they're deeper underground and they're much cooler. And so they moderate their body temperature through behavioral means. So they can maintain relatively constant body temperatures. They're not thermoconformers. Their body temperature doesn't conform to the environment around them, but they don't moderate their body temperature through physiology. They moderate their body temperature through behavior. The species I work with, though, is nocturnal. It's only active at night. There is no sun to go out and bask in. There's all the shade that they want because everything is shady because it's night, but they can't warm their bodies up, and so they tend to have body temperatures that are cooler, and in theory, they might have performance levels that occur at peak performance at cooler temperatures. But temperature varies seasonally, so if you are hanging out in a warm environment, you might find that your optimum speed is at a relatively high temperature. But then the seasons start to change and it starts to get colder. And during winter, when you're at colder temperatures, instead of having this kind of response of the relationship between performance and body temperature, you might change your physiology such that you perform better at lower temperatures than you would if you were being acclimated at warm temperatures. But the end result of this is that you can be active at lower temperatures than you could before, but your your optimum speed isn't as fast. So we might expect there to be some sort of cost in performance. And so that's something that we do. We go through acclimation every year. We This time of year, it's warming up. We begin to feel very warm, even though it's not all that warm outside, because we've been hanging out in cold temperatures all during the winter. As I speak about this right now, I'm warming up because I'm walking around and talking a lot. And so in the fall, when the temperatures drop, so what happens with students, something that I've observed every fall and every spring, October gets here, we get our first 50-degree day, all the students put on their sweaters and jackets and their long pants. And then they're in sweaters and jackets and long pants until spring. February or March comes around and we get our first 50-degree day. What happens? Everybody's in shorts and t-shirts. That's what acclimation is. We go through it. Lizards go through it. But we can also have an evolutionary response, such that if you're in a warm climate, you just perform better at warm temperatures and you suck at cold temperatures. And if you have evolved in a cold climate, you are not very good at really warm temperatures. Your peak is cooler, but that also means that you are capable of functioning at really cold temperatures. Both of these are strategies that organisms can use to kind of cope with changing climate. There can be an evolutionary response that happens over generations, or there can be an acclimation response that happens within individuals across the seasons of a year. So people have done work on this before. They found that body temperature at which, if you take a lizard and flip it over on its back, it can't flip itself back over. 10.5 degrees centigrade, the maximum is the high end at which you turn a lizard over on its back and it can't flip itself back over. In this case, it's because it's just too cold to move. In this case, it's because it's too hot that it's in the process of dying. These guys did not measure the preferred temperature, and I'll show you how we measure preferred temperature here in a few minutes. But they found that the preferred temperatures of geckos don't match the temperatures for maximum sprint speed. They run fastest at warmer temperatures than they actually prefer to be at, which is interesting because you would think that you would prefer to be at the temperature that you perform best at. Another study actually looked at preferred temperatures, found that there's a fairly wide range of preferred temperatures, but they only experience their body temperature in the preferred temperature during August at this particular site. But they only measured preferred temperature in the summertime. They didn't measure it in the winter. And one of the things that we might expect to see happening is preferred temperature changing seasonally if they're doing acclimation. Yeah?
What is defined as preferred for geckos?
You put them in a room where they can take on any temperature that they want, and then you come in periodically and measure their temperature. I'll show you physically what that looks like here in a few minutes.
Looks like that long trough, that's different.
Yep. Yeah. So I have a graphic. I think I've showed it to you before, but I'll go through it again. So hang on to that for a minute. We'll get to it in a few minutes. Good. So my questions are, is the sprint speed in this species changing as it moves out of southern populations into northern populations? And to try and get a handle on whether or not these changes are due to acclimation or to evolution. I have two sites that you're familiar with. University of Central Oklahoma, which is what I call the Oklahoma City site. It's just north of Oklahoma City. And then Galveston, Texas, which is down on the Gulf Coast. Galveston, because it's on the Gulf Coast, it stays warm all year round because it's next to this big warm mass of water. Like I said before, I work in skeevy places and nice places. But we check the body temperature of the lizard, which is what you guys are analyzing right now. We then measure the wall temperature, and then we take random selections of temperature all over the wall to quantify what the thermal environment is, because lizards can warm up by running out into the sun. These guys can't because they're only active at night, but they can actually press their bodies up against warm parts of a building or a rock or something and absorb heat through conduction into the body. So for example, at the University of Central Oklahoma, they have a bunch of buildings that we collect lizards off of, and then there's a physical plant building. It's a plant that generates the heating and cooling for the buildings around it. And in wintertime, what it's doing is it's heating up boilers, just like we have a boiler system that works here. And if you go up to that building in the middle of winter, it is to the touch warmer than all of the other buildings on campus. And if you're a gecko and you live on that building, chances are your body temperature is warmer simply because you are on the surface of the building that warms up the campus in winter. I was shocked at how poorly insulated that building was, such that the energy being generated in the building was actually radiating out through the walls of the building when I first touched it. So these are the environmental temperatures. And so we can know whether the animal is conforming to its environment by knowing whether its body temperature is reflective of the wall temperature right next to it and whether that is reflective of the wall temperature in general. Here's how you know what the preferred temperature is. You put them in a cold room, in a channel where they can't get out. There's a heat source at one end, but it's a cold room, so it's really hot at this end, it's really cold at this end. They position themselves wherever they feel most comfortable, and you just come through periodically and you check their body temperature, and you build up a distribution of body temperatures, and the average of that is their preferred temperature. Does that answer your question from earlier? Yeah. Then we put them in racetracks, and we race them. We designed a racetrack. We did the programming for it. It automatically records the data to a computer file. You put the gecko in at the beginning. When it runs through the first gate, it starts the timer. Second gate records the time between the first gate and the second gate, and then calculates that as centimeters per second. And you can see that in many cases they get faster as you go through. So they start out slow and then they build up speed and they're really screaming. This guy is running 2 meters per second at the end of the race. So these are quarter meter splits over a full meter track. Sometimes, these are all for the most part getting faster. This one slowed down at the end. It might have been a smaller gecko. Sometimes small geckos slow down more. We run these races at different temperatures. So we ran them at 10 degrees, 15 degrees, 20 degrees, 25 degrees, 30 degrees, 35 degrees, so that we could look at how body temperature changes their speed. So we collect them in the field. We put them in the lab at whatever temperature we collected them at. So in the winter, we put them in the lab at cold temperatures. We have a lab where we can control temperature to a certain degree. Then we will race them at a variety of different temperatures, and then we look at their preferred temperature in the thermal gradient that I showed you. Then we switch them to the other temperature. So if we collected them in winter, we then shift them to summer temperatures over about 3 weeks, and then we repeat the process. And if we collected them in the summer, we do this and this, and then we shift them over to winter temperatures over the course of about 3 weeks to acclimate them. Then we race them again, and then we measure their preferred body temperature. And then we just do a bunch of data analyses. And we're doing data analyses on all kinds of things. Sprint speed, using collection, acclimation, population, and temperature. So this is a 4-way analysis of variance. This is a 2-way ANOVA. No, this is a 3-way ANOVA on all of these different types of temperature. So this is what climate looks like. So from January to the end of the year, this is, I believe, 2018 data. The upper red line is the daytime high for Galveston. This is the daytime high for Oklahoma City. This is the nighttime low for Galveston. This is the nighttime low for Oklahoma City. The Oklahoma City geckos are always at cooler environments, at cooler temperatures than the Galveston geckos are throughout the year. The daytime highs converge during the summer, but the nighttime lows, it's always cooler in Oklahoma City. So because these guys are active at night, they're constantly exposed while they're active out on the walls. They're constantly exposed to cooler temperatures. So you might think that they might evolve to be better functioning at cooler temperatures. So there's a good reason to expect that that might be the case. These are the data that you are working on right now. So this is collection temperature, winter versus summer. The red is the Galveston population because it's warm. The purple or blue is the Edmond, Oklahoma population because it's colder. When we look at the winter collections, this is the environmental temperature on the walls. We see that Oklahoma City is cooler than Galveston. When we look at the wall temperature right next to the gecko, cooler temperatures in Oklahoma City than in Galveston. And when we look at their body temperatures, we see cooler temperatures in Oklahoma City than in Galveston. But when we put them in a thermal gradient and measure their preferred temperatures are not different from one another, which means that there probably isn't any acclimation, and there also probably isn't any sort of evolutionary response, at least where body temperature is concerned. These guys in Galveston are operating at temperatures that are pretty close to their preferred temperature. These ones in Oklahoma City are significantly below their preferred temperature, like way below their preferred temperature. But they're still crawling around on the walls. So even though their preferred temperature may not be changing, their performance temperature might be changing because they are active at much lower temperatures. In the summertime, everything converges because the environment in Galveston is much closer to the environment in Oklahoma City. And so the environmental temperatures are slightly lower, but it's not significant. The wall temperatures are slightly lower and the body temperatures are slightly lower. But once again, the preferred temperatures are not different from one another, nor are they different from the preferred temperatures in the winter. So preferred temperature isn't changing with these guys. At this point, you only know how to do a one-way ANOVA. So I have asked you to combine summer and population into a single variable because you only know how to do a one-way ANOVA. But I could also give you this entire data set. We filtered out, remember at the beginning, we filtered out environmental temperature, we filtered out wall temperature, we filtered out preferred temperature, because that would also require another factor. Today, well, maybe not today, but if not today, on Wednesday, we're going to start talking about a two-way ANOVA. A two-way ANOVA will allow us to look at collection season as one effect and population as another effect. So that's two factors, season of collection and population. We can examine each of those individually, and we can also examine how those things interact with one another. And you can, I think, probably already see how that interaction is going to work out because they're different during the winter, but they're not different during the summer. But if you can do a two-way ANOVA, you can do a three-way ANOVA. So we can actually look and see how season of collection and population affects body temperature, affects temperature, depending on whether it's environmental temperature, wall temperature, body temperature, or preferred temperature. So that's a three-way ANOVA. But a three-way ANOVA is mathematically just like a two-way ANOVA. There's just another variable there. Ultimately, what we're trying to figure out is, are these guys able to thermoregulate? One way to analyze this is by regression. So one of the techniques that we're going to do in this class is regression. If these things are thermoregulating effectively, their body temperatures will not be correlated with the wall temperature right next to them. But what we find is that the warmer the wall is, the warmer the gecko is. So these guys are not effectively thermoregulating, but this is not an analysis of variance. Now this is a regression analysis, which is the next thing that we will do after a two-way ANOVA. And then the last thing that we did was we actually erased these guys. And this is a really complicated analysis of variance because it also involves analysis of covariance, because geckos that are bigger run faster than geckos that are smaller. So we have to regress gecko speed on gecko length to get the residual from that analysis. That's then what we use to analyze the data. But what we find is that the populations differ depending on when they were collected and how they're acclimated. And in some cases, one population is bigger than the other. In other cases, they are the same. And in some cases, one population performs better at cold temperatures and another population performs better at warm temperatures, which once again might indicate this acclimation response that we see. Another thing that we learned in the process of doing this is that the populations are also different sizes. And so this is the two populations in winter. You'll see that there are a bunch of geckos that are just bigger than these geckos in Galveston. So Oklahoma City tends to have larger geckos. They're larger at adult size, and they have more weight per unit length. Once again, this is a regression analysis. So This is a data set that in order to understand it fully, you have to apply analysis of variance, you have to apply regression analysis, you have to apply analysis of covariance. It's not just one thing that's going to get you through it. And that's the reason that I concentrate on this particular analysis, because over time you kind of get to know the analysis, and it's a set of data that require multiple ways of looking at the data to answer different kinds of questions about the data. So your regression assignment is basically asking the question, are these animals thermoconformers or not? And what's the statistical support to indicate that they are or they are not? And so you're testing to see, is this slope of this line different from a slope of 0? If it's positive, then they're basically body temperatures are tracking environmental temperatures. And if it's flat, it means that their body temperatures are not tracking environmental temperatures, which would indicate that they're regulating their body temperature effectively. Regression analysis, okay? And these are exactly the kinds of projects that people will walk up to you with. And if you don't know anything about the thermal biology of geckos, you, the researcher, you, the data analysis person, the data scientist, have to learn enough about it to analyze it effectively. You don't have to know everything about it, but you have to know enough about it to analyze it correctly. Does this help with understanding why you have a body temperature data set that you're working on right now? Okay, good. That's what this is intended to do. After we learn two-way Anova, you're going to get this whole data set in all of its fullness: population, season, temperature type. Three-way Anova rather than a two-way Anova, but I'll show you when we go through two-way Anova, it's really no different in terms of the math. It just has another interaction term, another set of interaction terms. All right, questions? All right, we really don't have enough time to get started on two-way Anova. There is a two-way Anova PowerPoint on the Moodle site. Also, there is a whole chapter of your book that deals with two-way ANOVAs. In particular, I want you to explore the difference. Listen to me. You guys are ready to go, even though it's still like 7 minutes early. Explore the difference between what they mean when they talk about a replicated two-way ANOVA versus an unreplicated two-way ANOVA. We will cover those two things separately, but they differ in important ways, so read that in your textbook in preparation for Wednesday. If you have questions about the analysis you're currently working on, email me, come by my office, make an appointment, whatever, and I'll give you a hand with whatever you're having difficulty with. Yes, ma'am. I can be. What time do you want to come by? I don't have anything scheduled, and so I don't generally come in unless I have an appointment. I prefer getting things out of the way in the morning. Yeah, are you free in the morning?
Well, I'm done with class at 12.
Okay.
Just come right after.
I have another appointment at 5. Can you come at 4?
Yeah.
Okay, I'll see you at 4 then. Send me an appointment request. Yeah.
Are you, so you said you prefer to meet in the morning?
I do prefer, because then I have bigger blocks of time in the day.
I have class all day tomorrow. I'll lay in the morning tomorrow.
Okay.
What's your preferred time then?
3.
Oh, I did it in the morning though, because I'll be busy afternoon.
Oh, you have nothing.
In the morning.
Oh, you have nothing. I thought you said you had a full morning.
No.
Oh, I can meet you at 8.
Can we go like 10?
No. You have a full morning free?
Yeah.
Get your ass out of bed and eat breakfast and come and see me at 8.
8:30?
Okay, 8:30.
All right.
Man, I didn't realize this was a bargaining thing. You want something out of me and you're going to bargain? Yeah, 8:30. It just frees me up. I like to have big blocks of time to get stuff done. Yeah.
Thank you.
Sure. Send me a request also, just so it's on my calendar. Just make an appointment in Outlook. We all have access to Outlook. Oh, I can show you how. Go to Calendar.